Hi, this video is about the concept of D value and Z value in controlling growth of microbial spoilage. This is our group mate. The first one is me myself, Najwa Umairah, Chong Munian, Sajvini, Nur Afika, Nurul Naziha, and the last one is Nur Ifa. Definition of Z and D value.
D value is the amount of time to take to kill 90% of specific microorganisms in a given medium at a specific temperature. That means, from the D value, we can easily calculate the processing time required to achieve sterilization. Z value is temperature change that required to achieve reduction in the D value by a factor of 10. That means, Z value relates to the resistance of microorganisms at different temperature.
Difference between Z and D value.
As the definition of D value talks about time, so the measurement is time taken and the unit is minute. D value can be calculated directly from the graph of number of survived microorganisms versus time. So the formula is stated there, where N0 is initial bacteria load, while NT is number of bacteria survived after heat treatment. For Z value, the definition states about temperature, so the measurement is temperature change and the unit of Z value is Celsius. Z value can be calculated directly from the graph of log D versus time. So the formula that we can use is stated there.
Food type milk.
Content in milk are mostly water, which take out 87% of milk, especially for cow milk. Water can help to maintain blood volume, blood pressure, and body temperature. Water also helps in transportation of nutrients and waste in human body. Milk also contains 3.4% of milk fat, which composed of cholesterol and fatty acid. 65% saturated fatty acid, 29% monounsaturated fatty acid, and 6% polyunsaturated fatty acid. The polyunsaturated fatty acid include essential fatty acids such as linoleic and linolenic acid. Cholesterol are important components of cell membrane, while fatty acid can act as solvent for fat-soluble vitamins A, D, E, and K. Next, there is 3.3% of protein are found in milk. Milk protein contain all the essential amino acids and are composed of 82% of casein and 18% of whey protein. Proteins act as building block of muscle, skin, hair, and cellular components, synthesis enzymes, hormones, and antibodies, and also act as source of energy. Next, 4.9% of carbohydrates in milk are present in the form of lactose, which is the result of combination of glucose and galactose. Carbohydrates act as sources of energy and are important in hormonal regulation. Besides that, milk also contains many minerals and vitamins. Examples of vitamins are vitamin A, B, C, D, E, and K, whereas examples of minerals are calcium, copper, iron, magnesium, potassium, sodium, and others. There are also other minor biological proteins, enzymes, and hormones. However, these components does not have significant function to our human body.
Processing method.
So pasteurization is a relatively gentle heat treatment that involves heating of food to around 100 degrees Celsius. Pasteurization is used to increase the consumer milk safety by eliminating disease-causing bacteria, which is known as pathogen, and it helps to improve the shelf life and quality of the milk products. So these microorganisms that can withstand pasteurization temperatures, which are used in milk, are divided into two groups, which are known as thermoduric and thermophilic. So pasteurization has a lot of methods, but the most known methods have two, which are batch holding method and also high temperature short time, which is known as HTST. So in the first method, which is the batch holding method, every particle such as milk must be heated to at least 63 degrees Celsius and kept for at least 30 minutes in this manner. Whereas for the HTST method, in this process, each particle of the milk is heated to at least 72 degrees Celsius and held for at least 15 seconds. So then it is carried in a continual manner using UHT, which is the ultra heat treatment, a sterilizing procedure which may also be done at a greater temperature for shorter period of times. So these are the methods used to reduce the spoilage and also increase the shelf life.
Growth factor of microorganisms in milk.
We also know milk has one carbohydrate, which is sucrose or sugar. Sucrose can utilize sources of energy more quickly than fat and protein. However, the energy currency of the cell is glucose. So, to use available carbohydrate, microorganisms must be able to convert them to glucose. The only sugar naturally present in milk is lactose. Most microorganisms lack in the enzyme lactase, which is required to break down lactose into two component sugar, which is glucose and galactose. The lactic acid bacteria, which do have the lactase readily, break down the lactose and use glucose as energy sources. Lactic acid bacteria therefore have a competitive advantage in milk. They are able to grow bacteria which are unable to obtain glucose from lactose. Further, some lactic acid bacteria are able to convert galactose to glucose. Secondly, the acidity or pH. The titrable acidity of milk typically varies from 0.1.2 to 0.19% lactic acid depending on composition, especially protein content. The pH of milk is near the psychological of pH of 6.8, which is considering the following points mean that the milk is a good growth medium with respect to the acidity or pH. The major group of microorganisms important to preservation are in order of increasing acid tolerance, which is bacteria, yeast and molds. Natural fermentation of warm raw milk by lactic acid bacteria reduce milk pH to less than 4.0, which is prevent the growth of pathogenic bacteria and most spoilage bacteria. Lastly, the factor of growth microorganism in milk, which is a moisture. Milk has a higher moisture content, which is 87% for cow milk, and with respect to available moisture, is an excellent growth medium. But it must be understood that with respect to microbial growth, the critical parameter is water activity, not the moisture content. Water activity, or the AW, is an index of the availability of water for microbial growth. Microorganisms vary greatly in their ability to survive. For example, most bacteria can survive in 0.90 until 0.91. The most yeast can survive 0.87 to 0.94. The most mold can survive at 0.0 to 0.80.
Importance of Z and D value in food industry.
Z value. The increase in temperature required to reduce D to 1/10 is value or to reduce. Second, the spores killing effect of heat treatment can be expressed as a function of temperature, the time the material has been exposed to that heat, which means heat treatment are easy to killing spores. D value. To reduce the number of microbes in product to ensure proper food safety. This is done by thermal processing and ways to reduce the number of bacteria in the product. Second, the extent of the pasteurization treatment required by the heat resistant of the most heat resistant enzyme or microorganism in the food. Third, the heating time required to process the product will also be increased. Fourth, UHT milk processing, very high temperature for very short time compared to lower temperature, longer time process. If the product is heat treated at a temperature of not less than 135 Celsius for a short time, aseptically packaged in sterilized container has minimum chemical, physical in relation to the severity of the heat. Thank you.